in this particular lecture, we are talking about the heart in the case of fishes. Now, students, in the previous part of the lecture, we had talked about the heart of the lamprey, and we have seen that we had two chambers in that particular case. So, the first chamber was the auricle, second was the ventricle, and there we had two accessory chambers, which were the sinus venosus and the conus arteriosus. So, as a whole, we have got a two-chambered heart with two accessory chambers right now. And now we will see that what is the case of fishes. Now, in this particular case, the number of the chambers is quite similar as we have seen before. Now, if I go further into this part, so this is a heart of Squalus acanthias. So this is the dogfish heart. And if we see you can say the anatomy of this particular heart, we have an atrium, which is very comparable to the heart of the lamprey, as we have seen before. So this is the atrium. We have the second chamber as the ventricle. So these are the two chambers which are being discussed earlier also. Now here you can very well see that the atrium, it is on the lower side and the ventricle is over it. That means they are not anterior-posteriorly placed, but they are one on the other. The ventricle is present on the top of the atrium. So this is a kind of placement in this particular heart. Now, if we go further, we have again one accessory chamber, which is the sinus venosus. So this is the accessory chamber which is also present in this case, in the case of fishes. And this chamber is serving to store the blood which is coming from the body. And as we have previously discussed, in these types of hearts, we have only and only the deoxygenated blood coming to the heart. So that's why it is also called as the venous heart. Now, if I go further, we have again a conus arteriosus. So that means these are very similar kind of hearts, but the modification is that you have so this heart is bigger than the previous one. So this is a size modification that you can see in the fishes. Now, after seeing the particular structure of this particular heart, so this is a typical two-chambered heart, and this is the case of the fishes. So in fishes, we have got the two-chambered heart with two accessory chambers as the sinus venosus and the conus arteriosus. Now there are some other modifications which we will be taking on later on in the conus arteriosus. What are the number of the valves, how these are arranged. So these are some of the modifications that have been seen in the case of fishes as compared to the previous heart type. Now if we go further in fishes, this heart is of the Squalus acanthias. This is the dogfish. Now here we have a single circulation heart. Now, as we have previously discussed also, that the deoxygenated blood from the body will be coming into the sinus venosus, and it will be stored for some time. It will be given to the auricle. Then it will be pumped to the ventricle, and through the conus arteriosus, it will be going into the ventral aorta, which is present on the top. I should say it is in the anterior side. As the heart of the fishes, if you see the position of this heart, it is ventral in the body. So if you have a fish, it will be present on the ventral side of the fish. Now this ventral aorta, it will be going anteriorly towards the head part. And these are the efferent branchial arteries which are coming out of the ventral aorta, and these will be going to, as they are branchial arteries, so they are going to the branchia, they are going to the gills. Now why they are going to the gills? Because the deoxygenated blood from the body, it is coming into the sinus venosus, then it is going into the auricle, then to the ventricle, then to the conus, then it is coming to ventral aorta, then to the gills through these efferent branchial arteries for oxygenation. Now, at the gill section, at the gill level, it is oxygenated, then it goes to the head part or to the body for, you can say, the transportation of the oxygen and the food. So this is the mechanism that how it works. So that's why they are called as the single circulation hearts or the venous hearts. Now if I go further, so there are different arrangements of the semilunar valves which are present in the conus arteriosus. Now what is the arrangement? So what are the arrangements which are seen in the case of fishes? Now if I go for the first case, these are numerous in the elasmobranchs. So that means their number is huge in case of elasmobranchs. So these are the cartilaginous fishes. So we got many semilunar valves in the case of elasmobranchs. Now if I go further, these are arranged in 3 rows. Now here you can very well see, if you see the diagram, here you can see that these are arranged in 2 rows. I should say 3 rows. So how these are arranged? That 1 row is dorsal and 2 are ventrolateral in position. So here you can very well see this is the dorsal row of the valves and 2 are the ventrolateral rows of the valves. So this is the arrangement of the valves in case of elasmobranchs. As this is a dogfish, so we are talking about these kinds of hearts. So here we have the semilunar valves in 3 rows. Now if I go further, the valves of the anterior set, they are larger in size. So here in the diagram also you can very well see, if you see this row of the valves, which is the anterior row, or you can say the anterior set, they are larger than the posterior set. Now this may be attributed to a higher efficiency to make higher pressures when the blood is going out of the conus arteriosus. Now these valves, they are helping in to check the backflow of the blood into the ventricle, and the second thing is that they are maintaining a pressure, or they are helping the conus arteriosus to maintain a pressure when the blood is flowing into the ventral aorta through this set of valves. Now here is an example. This is you can say another example. In the case of Neoceratodus, we have got 8 sets of these valves, of these semilunar valves. Now if we go further, there are many elasmobranchs in which you have 6 sets. So as we have discussed earlier, that the elasmobranchs, they have numerous, you can say, the semilunar valves, but here it is 6 sets. They can be 6 in number. Neoceratodus has got 8 sets, and if we go further, the Tediosts, they have managed to cope up with this particular pressure kind of condition with only a single set of semilunar valves. So there are only a single set of the valves present in case of the bony fishes. So Tediosts are the bony fishes. Now these are some of the arrangements of the semilunar valves in the heart of the fishes. Now if we go by the evolution, if we see the evolution of the fish heart, what is the tendency in fishes? Now, if we go by the evolution, what is the tendency? It is the reduction in the number of the valves. So, in the primitive kind of fishes, if you see the Neoceratodus or the primitive elasmobranchs, they have got large number of numerous set of valves, of semilunar valves. But as the evolution it progressed, the newer, the modern fishes, they have got a reduced number of valves, as we are seeing in the case of TDOS. So they have got only one set. So this is the tendency in the reduction of the number of the valves in the case of fishes during evolution. Now students, after this particular thing, I'm just giving you a concept that how the heart is present in the fishes and how the circulation occurs in the case of fishes. Now in this particular diagram, you can very well see the position of the heart in the fishes. Now this is the position of the heart. And here we have the four chambers, or you can say the two main chambers and the two accessory chambers of the fish. We have got a sinus venosus, we have got an atrium over the ventricle. So this is the ventricle, which is pumping, which is the muscular organ. And this is the conus arteriosus. So this is the particular ventricle heart, which is ventral in position in case of a typical fish. Now, how it pumps blood? That from the body, the deoxygenated blood is coming to the sinus venosus. It will be coming to the auricle. It will be going to the ventricle through the auriculoventricular valves. And from the ventricle, it will be going through the conus into the afferent branchial arteries, as we have seen in the previous diagram. Now from these branchial arteries, they are going into this seat of oxygenation, which are the gills. So when the blood passes through the gills, it is oxygenated over here, and what we have on the top, it is the dorsal aorta. So from this dorsal aorta, the blood may go to the body, to the whole body behind, and to the head part, to the brain in the anterior part. So after the transportation of the gases and the food, the whole deoxygenated blood, it is coming back to the heart, then it is pumped through the gills, then it is going back to the body. So this is the course of circulation in case of fishes. And here you can very well see that the heart is receiving only and only the deoxygenated blood. So that is why they are called as the venous hearts. Now if we go further, this particular diagram is giving you an idea that how the heart is present in the fishes. So this is the original dissected, you can say, diagram of the heart in the fishes. So this is the heart proper, which is present. And if we go further of the heart, we have this conus arteriosus. So this is the ventricle, this is the conus arteriosus, then we have the ventral aorta. This is the ventral aorta. And after dissection, so what we have done is that it has been injected with a blue dye. So from the heart, the deoxygenated blood is going upwards into the ventral aorta. So these are the branchioles, the afferent branchioles, into which the deoxygenated blood is going. And here you can see the presence of the gills. So these are pink colored gills. So it is going for both the sides, onto the left and the right side, and it is getting oxygenated. Now if we go for the next diagram, this is the next figure. These are the injected efferents. So from where we have the blood going to the gills. Now if we see this particular picture, Now here you can see the oxygenation process. So from the heart, that was the seat of the heart, the blood was coming into ventral aorta and it is going to the gills. Now these are the gills. You can very well see these are the lamellae of the gills. And in these seats of oxygenation, the blood is oxygenated. It becomes red in color. These are the efferent branchial arteries. And from here, they join the common dorsal aorta, which is this particular dorsal aorta. So these are showing you the injected efferents. So here we have the efferents, here we have the efferents. And now I think you can grab upon the idea that how the oxygenation process occurs in the fishes. Now students, there is a special case you can say in case of fishes. Now, what is this particular case? Now, before starting this particular slide, I would like to give you a concept that fishes have got two-chambered heart. Up till now, we have studied a two-chambered heart, and they have got two accessory chambers. One is sinus, and second is the conus. Now here, we are taking the case of the dipnonian fishes. So there is a group of fishes which is called as dipnoi. Now these are also called as the lung fishes. Now as the name suggests, the lung fishes, they will be having lungs or a single lung with the gills. So there will be both the organs, and there are two seats of oxygenation in these kind of fishes. So we have gills also, we have got the lung also. So we are also saying it as air bladder. So these are the dipnonian fishes in which we have to see now that what is the modification in the heart if we have got a lung in spite of the gills. Now, here what we have studied till now, that the fishes, they have got two-chambered heart till now. But in dipnonians, we have got a typical three-chambered heart. Now this is a modification, this is the evolution of the heart. So if you have lungs in the fishes, you have got three chambers. If you have got gills, you have got two chambers. And here we have a gill and a lung, so we have got three chambers. Now we have to see that why they have got a three-chambered heart. Why they cannot take it with a two-chambered heart. So dipnonians have got gills and lungs. So why they have developed a three-chambered heart? This is the question. So we have to see the structure of the heart for this particular question. And the answer to this is that in dipnonians, what has happened, that the atrium, it has been divided into two parts. We have the right atrium and we have the left atrium. And we have a single ventricle, as in the case of these fishes. So here we had one auricle, one ventricle, but in dipnonians, we have got two auricles and one ventricle. So that's how we have got a three-chambered heart in the case of dipnonians. Now students, the question arises that what is the need to make a left and a right atria? Now this is in correlation with the formation of the lung in these fishes. So we'll be seeing this particular thing in the later slides. Now, the atrium is divided into right and left atria, but this is the most important thing. We have a partial septum in between these two atria. We have right and left atria, but there is a partial septum. That means this septum is not complete. So this is not a true kind of three-chambered heart. So we can write it as a 2.5 chambered heart. 2.5. That means this partial chamber is the partial septum, which is present in between these two atria. It is not full. And if it is not full, what can happen? So there can be some mixing of the blood in between these two atria. But that mixing uh, is hindered to some extent by some other methods. So I'll be taking those methods right in the coming slides. Now students, if I show you the diagram of a Dipnonian heart, so here you can very well see the auricle, it has got a partial septum inside it. As you can very well see, this is the partial septum. This is the ventricle. So we have got 1.5 auricles and 1 ventricle. So it can be 2.5 chambered, but it has been written as a 3-chambered heart. Now students, I am taking one example of the Prototeris. Now this is a Dipnonian kind of a fish. So it has got a venous heart, first of all. So deoxygenated blood is coming to this particular heart every time. And it is containing the unoxygenated blood from the body. Now, as we have discussed earlier, so it will be taking the unoxygenated blood or the deoxygenated blood. So it is going for the right atrium through sinus venosus. Now this is the most important thing to note, that the unoxygenated or deoxygenated blood from the body, it is going for the right atrium, not into the left atrium. Now this is a separation. This is the modification. Now here, I want to emphasize on this particular point that what we discussed earlier in the case of fishes, that the deoxygenated blood from the body, it first of all comes to the sinus venosus. Then it is stored for some time. Then it goes to the auricle. Now here, the sinus venosus has got only the contact with the right atrium. Sinus venosus has got no contact with the left part of the atrium. So this is the modification. Now if we go further, these fishes, they have got a swim bladder also. So this is the lung. This is the seat of another oxygenation seat. So if we have the swim bladder, what we have, that the oxygenated blood from the swim bladder, it is coming to the left atrium. Now this is the mechanism. So you should be very clear in this particular part that we have got two parts of the auricle, left and the right. It has got a partial septum. The right part is having the deoxygenated blood from the body through sinus venosus. The left atrium has got the oxygenated blood from the swim bladder. So that means the two auricles, they are containing different types of blood. So if we go further, there is a partial septum. So there can be little mixing of the blood of left atrium and the right atrium. So there can be some mixing in the auricles. So little mixing occurs, but there is a mechanism. So two streams are kept separate. So these kinds of fishes, they have got a tendency, their hearts, they have got a tendency to keep these two streams separate, though they have got partial septum. Now how they keep these streams apart? It is with the rugae-like structures which are present in the two auricles. So these are the infoldings and the outfoldings which are present in the wall of the auricle. that are preventing some of the mixing in the two auricles. Now students, this is a particular diagram which is giving you a concept that how the mixing or the partial septum is going for its particular mechanism. Now, what we have in the dipnonians, we have got a left atrium, we have got a right atrium, and we have got a single ventricle. The right atrium, it is receiving the deoxygenated blood, as it has been shown over here as blue square. The left atrium, it is receiving the oxygenated blood from the swim bladder or the lung of these fishes. The ventricle is present over here. Now students, we have a sinus venosus and a conus arteriosus. So these are the two accessory chambers present over here. So as we have discussed earlier in the slide. So what we have now? We have got a partial intra-auricular septum. So I have taken a black color for this. So this is a partial septum in between the right and the left atrium. And what we have is that the sinus venosus, it is in contact with only the right auricle. So there is no connection of the sinus venosus with the left atrium. So that's why the left atrium has got the oxygenated blood and the right atrium has got the deoxygenated blood from the sinus venosus. Now, if I go further, the ventricle is also partially divided. So this is a kind of modification. Now, why it is partially divided? To keep the two streams separate. Now, if we have the blood going from the left atrium into the ventricle, and we have the blood coming from the right auricle into the ventricle, so there is complete mixing. So there is no, you can say, advantage of making these two auricles. So we have a partially divided ventricle also. So this is a partial, you can say, septum present in the ventricle. So it is keeping the two streams separate to as much as it can prevent. Now we have the conus arteriosus with a partial septum also. So this is I think this is the height of the efficiency. This is the height of the modification, or you can say this is the height of the evolutionary phenomenon, which is automatically making these partial septums in the heart of these kinds of fishes. Now, to cope up with the mixing error in these hearts, we have got this septum, we have got a septum in the ventricle, we have got a septum in the conus arteriosus. So what is the result? The stream from the lungs, it is going for the auricle, the left auricle, it is going for the left part of the ventricle, and then it's going to the left part of the conus arteriosus, and it is leaving the heart into the ventral aorta. Now, the aorta, it is again subdivided into different arteries. So I'll be taking those arteries in the coming slides. So if you go for the right atrium, we have got the deoxygenated blood, which is coming from the sinus venosus, and from this right auricle, it is going to the left or the right part of the ventricle, it is going to the right part of the conus, and it is going to the ventral aorta. Now students, I think you are very much clear about this. Now here we are showing that the stream on the left side is kept separate from the stream on the right side. So this is a kind of double circulation. So students, in these fishes, we have got the first signs of the double circulation mechanism, which the humans also have. We have got the double circulation. So in these fishes, This is the first, you can say, starting point of double circulation, though we have got some errors, but still we have got double circulation. Now this is a particular picture which is showing you the flow of the blood in these fishes. Now I start with the circulation in the lung fishes. So what we have in this particular diagram, that you have an atrium which is divided into two, right and the left atria. So here you have the two atria. We have the ventricle, we have the you can say conus arteriosus. Now here it is labeled as the bulbous cordis. Now in these lung fishes, many of the books, they have termed it as the bulbous cordis. So you may get a question on bulbous cordis. So this is again, that is you can say a modification of conus arteriosus. Now this is quite big in size as compared to the conus. It has got a septum. Now here you can very well see the blue color is for the oxygenated blood and the gray color is for the deoxygenated blood. So the oxygenated blood, it is coming to the auricle from the lung. So as this is coming from the lung, it is coming to the auricle. So from here it is going to the ventricle, keeping the two streams separate, and the deoxygenated blood is coming from the body, and these two streams are kept separate in all the chambers. Now here, one thing is missing, that we don't have a sinus venosus over here. But you can put a sinus venosus in contact with this part of the heart, from where the deoxygenated blood is going into the auricle. So what is happening, that the oxygenated blood from the heart, it is going into the dorsal aorta straightforwardly, and it is going into the tissues. The deoxygenated blood of the body is going through the heart into the gills for oxygenation, and after oxygenation there is a special kind of duct which is called as the pulmonary vasomotor segment. Now from here the oxygenated blood is flowing into two directions. Number one, it can mix with the dorsal aorta into the oxygenated blood, and it can move to the lung also for further oxygenation. So after a kind of double circulation or double oxygenation mechanism, so you can very well see that deoxygenated blood from the heart, it is going first into the gills, then it is going into the lung for double oxygenation, then it is going for the heart. So this blood in the heart is quite pure after the double kind of oxygenation in these kind of fishes. So students, that was about the circulation of the blood in the lung fishes. Now there is something which is called as the coronary circulation. Now what is coronary circulation? This is the circulation which is needed by the heart itself. So this is the circulation of the heart, because the muscles of the heart, they are always dependent upon the blood to work. So the heart is also needing some blood for its own working. So this is the coronary circulation, which is for the heart itself. So in case of fishes, these are the efferent branchioles from which the fresh oxygenated blood is flowing in these particular efferent arterioles. This is what we know. Now, there are some hypobranchial arteries which are arising from these efferents. Now, as the blood has to go to the heart itself, so that should be a very pure blood, and that is taken from these efferents. Now, from these efferents, hypobranchial arteries, they arise. Now, what happens? These are the efferents from which the hypobranchial arteries they arise. Now, these hypobranchial arteries, they branch into many. So they are ramifying, they are branching into many arterioles. So they are forming the coronary arteries that will ultimately go to the heart, and they will be supplying the wall of the heart, the muscles of the heart to work. So that is the case of the coronary circulation in case of fishes. So in the next part, we will be taking up the amphibians into account. So that was all about the case of fishes. We have got a typical heart of the fish, which was two-chambered. Then we talked about the heart of the dipnonians. Here we have got a gills, we have got a lung also. So in these cases, we have got a three-chambered heart. So why it is called as three-chambered? Because we have got two auricles and a single ventricle. But here we have seen that the heart was also partially divided. All the chambers were partially divided. So we also discussed the course of circulation, and we also discussed the coronary circulation in case of the fishes. So students, with this we finish with the circulation in case of fishes.